Namaskaram students. In the previous session, we are discussed about chromosomal theory of inheritance, in which we saw the salient features, historical development, and so on. If there is any changes in the chromosomal structure, what would be happen? Ultimately, it leads to mutation. That's the topic today we are going to discuss. Mutation. It is a sudden change in the genetical material of an organism or sudden change or any alteration in the genetical composition of an organism leads to mutation. For variation, there are two important processes essential that leads to the evolution. They are mutation and recombinations. So these two processes which are essential for the process of genetic variations. And today we are going to see mutation topic alone. I already said a sudden change in the genetic material of an organism is called mutation. This term was coined by Hugo de Virus in the year 1901. He worked on the plant evening primrose. We have to call the name is botanically Enothera lamarckiana. So based upon his work, he proposed a theory name is called as mutation theory. So mutation and recombination is responsible for genetic variation. That's the ultimate source of evolution process. So any agent which causes the mutation are called as mutagens. The production of the mutant can expose to the different types of mutagens is called as mutagenesis. This is nothing but agent causing the mutation is called as mutagens. The process is called as mutagenesis. The organisms are exposed to mutation. So that organism we have to call the name is mutagenized. Just observe the picture. We know the carrot. Let's see how the characters appear here. And the types of leaves and a dahlia flower as well as a sunflower. Two variety of colors to be seen here. So these are the mutagenized organism. And next we are move on to how these mutations are occur. This mutation either to be a nature or it should be a artificial. So if it is the artificial means as a biologist or any technological persons, we are using some of the rays to induce the mutations. The rays such as X-ray, gamma ray, UV ray and some other electromagnetic radiation. For example, in this type of mutation, natural mutation is occur here with the help of the gamma rays or X-rays. Here, this shows the effects mutation rate 1000 to 1 million time increases than the normal effect. So, such a type of mutagenized materials we have to call the name as irradiated plants. But this mutation it has some of the optimum or normal level. If the mutation level is high, it leads to the death of the organism. If the mutation is very low, the exposure of the mutation can take longer time of the duration. And it is essential for germination and growth of the plantlet also. If the mutations are very minute, they are in the recessive condition means it is very invisible and it can be expressed generation after generation. We have to know whether the mutation is occur or not. It's art of mutational breeding. So that breeding purpose persons involved in the genetic engineering alone, they know whether the mutation is occur or not. Just observe the citrus fruit. Before the mutation, we can see the different types of ovules that is called as a multicarpellary. From each carpels, one seeds are emerges out. But after the irradiation materials, we can see there is no seeds. So these are the examples for how the irradiations to be working. And next we have to see about what are the effects of nuclear radiation. In our country, we heard about Bhopal gas tragedy. There is one chemical industry. From that, the gas called the name is methyl isocyanate, which is released and causes so much of drawbacks effects still now. Same thing which has happened in Russia also. The effects of Chernobyl of nuclear radiation. As a result, it affects not only the plant kingdom, it can affect both the animal as well as the plant kingdom. Just observe the pictures. Whether they are the normal? Certainly not. So this shows some of the deleterious effects in the mutational process. So this type of mutation is not happen afterwards because of the proper disposal of the radiation from the industries, which was monitored by the nuclear power plant projects. So these are the examples of mutational process, and these are the deleterious effect or causes the damages to the environment. Such type of progeny, they are not be fit for the environmental. After few days they are going to be destroyed. And next we are move on to the what are the other radiation is responsible for the mutations. Mutation, if any plants, if it is exposes to the UV radiations, just observe the pictures, there is a bond in the breakages. In the UV double-stranded structures, there is a slight bond breakage is observed here. When the exposure of the ultraviolet photon, it causes the in between the bond region, hydrogen is going to be a break. As a result, the pair of the genes to be a move apart from the normal position. So we know about thymine is always paired with the adenine. But here in the pyrimidine dimer, instead of thymine is joined with the adenine, here thymine itself they are going to be joined. As a result, a small bend to be occurs in the structure of DNA. Such type of a bend is called as a kink. So a kink means a turn or bend made in the DNA. Actually it should be in the straight position, but due to the exposure of the ultraviolet radiation, this bond, it is instead of joining with the adenine, thymine, they are joined with thymine itself. This is also the mutation because of the exposure of UV radiations. And next we are going to see about what are the mutational breeding results so far. So in the Chinese government, they are exporting the seeds to the space for the exposuring the radiations to promote the plant mutation. So such type of mutation, if it is a good, that alone they are come back to the earth and producing the yield. So for example, eggplant, brinjal, chilies and snapdragon. This shows when compared to the normal plant, they are increases in the height, yield also faster and the quality also better. But they are producing the seed which are very very low in number. So if any coin have both positive as well as the negative. So in the mutation, if it is a positive, we are going to be continue the process. If it is a negative means we are discontinue it. And next we are going to see about what are the types of mutation. So mutation based upon the origin, we are divided into two types. They are spontaneous or induced. Spontaneous means without any agent, they itself they are doing the process is called as spontaneous. Whereas induced means because of the presence of any agents. That agent may be either physical, previously we discussed about X-ray, UV ray or gamma rays. If it is a chemical means some of the chemical agents. So by using these, we are inducing the process. So we have to call the name as induced mutation. And second, based upon the cell type. Any living organism, we are having the two types of cell. One is called as a somatic cell and another one is a germline cell. If it is the somatic cell means, we have to call the name as non-reproductive cells. If the mutation is occur in the body cell, we have to call the name as somatic cell. If it is occurring in the germ cell, we have to say the name is reproductive mutation. And what are the effects of this mutation? It is again divided into four types. They are, if it is loss of function. So if any mutation is occur, that's not causes any kind of the problem. It shows the normal function. This is called as a knockout mutation. And second type is called as a hypomorphic. That is exposure of the mutation in the very low level. We have to call the name as hypomorphic. This shows the effects such as it can smally reduces the normal function. If the exposure of the mutation in the higher dosage level, we have to call the name as hypermorphic level. It can increases the normal function. This is the effect of mutation. And last type is called as a ectotropic expression, in which the expression of the mutation it's not in the proper time. It can be shown in the appropriate or incorrect. It is an inappropriate or incorrect time it should be expressed. So these are the types of mutation based upon the origin, cell type and its effect. molecular gene and effects of translation we'll see in the later. And next we have to move on to the types of mutation. There are two types of mutation. One is called as a gene mutation and another one is the chromosomal mutation. If the mutation is seen in individual genes, we have to call the name as gene mutation. It is also called the name as point mutation. Again I am saying if the mutation is seen in the single gene or individual gene is called the gene mutation or point mutation. If the mutations seen in the number of structures or chromosome is going to be changed, such type of mutation is called as chromosomal mutation. If any changes in the structure or number of chromosome, we have to call the name as chromosomal mutation. Then this joint gene mutation or point mutation again subdivided into two types. They are substitution or insertion or deletion. In the substitution, again it is subdivided into two types. They are transition and transversion. Let's see the one by one. What is the gene mutation? Gene mutation or point mutation. This is also called the name of substitution mutation or indel mutation. So this type of gene mutation again subdivided into four types. They are synonyms or silent mutation, non-synonyms or missense mutation, and third type is the nonsense mutation, and the fourth one is frame shift mutation. Let's see the first one. Silent mutation. What's the name of silent? That means nothing to be happen. Here, if you are changing any codon, what is codon? Three genes join together to form one amino acid. So that is called as codon. If any changes in the codon, there is no effects in the amino acid production. That is called as synonyms or silent mutation. Whereas in missense mutation, any change in the codon of one codon, it changes into the another codon, that leads to the effects of changes in the amino acid production. Again I am saying if it is the missense mutation in which one amino acid is changes into the codon for another amino acid. This is also called the name as non-synonyms or missense mutation. And third type is called as nonsense mutation. Nonsense, that means it shows some of the irritation. If any codon of one amino acid is changed, it can stop the process. Further mutation and further production of amino acid is going to be stopped. So such type of mutation is called nonsense mutation. And last type is called as a frame shift mutation. This is either the addition or deletion of the single base pair from one particular location to the another location is called as frame shift mutation. This leads to the changes in the DNA structure, RNA formation. This is called as a translation process. As a result, complete synthesis of the normal protein structure and function is going to be changes. So these are the four types of gene mutation or point mutation. And next we are move on to in detail. I'll explain again what are the mutagenic agents. I said about any mutation which is caused by some process is called as a mutagenic agents. There are three types of mutagenic agents. They are physical mutagens, chemical mutagens and biological mutagens. First we will see the physical mutation. Physical means some of the rays, temperature, heat, light. These are the different types of physical radiations. So under the physical mutation, again they are subdivided into two types. They are temperature and radiation. If the temperature is increases, it can break the hydrogen bonds between the nucleotide. As a result, both replication and transcription is going to be affected. And next type is called as a radiation. There are two types of radiation. One is called as the ionization radiation and another is non-ionizing radiation. This is a very important 5-mark question answer. Listen carefully, ma. Maybe one word twisted, one word will come from ionizing and non-ionizing radiation. If the wavelength are shorter and they have higher energy, this belongs to the ionizing radiation. Example, X-ray, gamma rays, alpha rays, beta rays and cosmic rays. So if any plants are affected by these kind of the rays, we have to call that plant is called as a irradiated plants. So as a result, it can cause the chromosomal mutation. So these are the effects of ionizing radiation. And next we are move on to the non-ionizing radiation. Here they have the longer wavelength and they are producing one of the lower level of energy. One example alone, UV rays. And this leads to mutation in the unicellular structures such as spore formation, pollen formation and microbials. So lower type of ionization radiation occur in the lower group of unicellular organisms, spores, pollen grains which possess nuclei which are occur at the surface membrane. And next we are move on to the chemical mutation. And chemical mutations, this is nothing but by using some of the chemicals we are inducing the mutation are called as chemical mutagens. Some of the examples are mustard gas, nitrous acid, EMS and MMS. EMS means ethyl methane sulfonate. MMS means methyl methane sulfonate. In addition to that, ethyl urate, manganese salt, formaldehyde, eosin and anthracene. So these are the examples for chemical using the mutagens. And next we are going to say very important chemical mutagens, nitrous oxide. So this nitrous oxide, if it is exposed to the plant surface region, it can change the nitrogenous bases of the DNA. As a result, it can disturb both replication as well as the transcription process. Ultimately, the production of protein, production of another process, polypeptide formation going to be interrupted in the translation process. And at last we are going to see about the biological mutations, in which maize commonly is an example seen in the transposition process. It is also called the name as mobile elements or jumping genes, in which some of the genes are going to be inserted by their itself. The presence of gene instead of one location, automatically they are going to changes in the another location, and this causes a mutation. Such type of mutation is called as biological mutation. We can see in the maize plants, and these are the example for the types of mutation. Next, some of the examples we are going to see some of the mutant varieties. The first mutant varieties are Sarbothi sonara. So it is a Mexican variety. So the name of the plant is called as a Sarbothi sonara. It is a mutant variety of wheat which is developed from the Mexican variety. The name of the variety is Sonara 64, and this variety is exposed to the gamma rays. This variety, which was worked by the person belongs to our country, Dr. M.S. Swaminathan. Commonly, famously we are called, the name is called as father of Indian Green Revolution. And second type is called as the Castor arona. Castor means Amanaku. It is a mutant variety of castor. Here the seeds are treated with thermal nutrients in order to develop early. If it is a normal day, this plant can produce the yield in 270 days. Whereas if it is exposed to the thermal, that is the heat nutrients, they can be produced within 120 days. So these are the example for mutant varieties. They are Sarbothi sonara and Castor aruna. And what is co-mutagens? Co means in addition. The compound which is not be mutagenic property, but it can be joined with some other compounds. It shows the effects of mutation. So these are called as the co-mutagens. For example, in ascorbic acid, it can increase the damages caused by hydrogen peroxide. Same thing in coffee, we can see the toxic substance called as a caffeine, so that it can increase the toxicity of methotrexate. So caffeine increases the toxicity of methotrexate. And in addition to that, we are going to induce some of the gases for our betterment of our product. So such type of mutation is called as a induced mutation. So that leads to cancer also. That's called a carcinogenes mutation. For example, if you are adding the alkylating agent, it can remove the normal base. If you are adding the aciridine dyes, it can either add or remove the bases. If it is frequently exposure to the X-rays, definitely it can break our chromosome, leads to the cancer. Similar to that, UV radiation also, it can create the thymine dimers. So previously we saw. So site-directed mutagenesis means that particular gene alone is going to be affected. And what are the facts which is related to the mutagens? If it is mustard gas, inside they are having the chemical called, the name is dichloroethyl sulfate. This is a powerful weapon which was used in the World War I to kill the major persons. And H.J. Muller in the year 1928, he was the first to use the X-ray to induce the mutation in fruit fly. L.J. Stadler reported the mutation in plants by using both X-rays as well as the gamma rays. So these are the chemical mutagenesis which was first reported by C. Arbach in the year 1944. So today I am summarizing the topic, ma'am. Today we discussed about the mutation, what are the types of mutation, in which we detailedly saw about the gene mutation and what are the mutagenic agents, and some of the example varieties of mutagenic products. Thank you, girls.